My name is Joyce Fung, and I would like to present to you a recent work in the Chook lab at UT Southwestern, which explains how Nap1 and Kap114 cooperate in H2A-H2B import and nucleosome assembly. During the S phase of the cell cycle, newly replicated DNA must be packed into nucleosomes in the nucleus. Core histones, which are rapidly synthesized and processed in the cytoplasm, must be imported into the nucleus. More than 20 years ago, Lucy Pemberton's group reported that H2A-H2B are imported by the importin Kap114. Histone chaperone Nap1 was found to bind to both proteins, but it was unclear how it affects the import and the nucleosome assembly process. This JCP paper is the fourth in a series of Chook lab papers on H2A-H2B nuclear import. The first paper by Padavaneal et al. In 2019 shows how importin 9, the human homolog of Kap114, recognizes the histone. It also revealed that RanGTP can bind to form an unusual stable ternary complex. The next two papers, Zhou et al. And Schaefer et al. In 2023, the latter led by the Darcy lab, used Cryo-EM and hydrogen deuterium exchange to reveal the structure of the ternary RanGTP Kap114 H2A-H2B complex. Finally, our latest paper presents biochemical and structural analysis of the three proteins in the presence of Nap1 and revealed their role in nucleosome assembly. We first used pull-down binding assays and secmoles to show that Kap114, H2A-H2B, and the Nap1 dimer form equimolar complexes in the absence and presence of RanGTP. We solved the 3.5 angstrom resolution Cryo-EM structure of the ternary Nap1 histone Cap complex, which likely represents the cytosolic form of the import complex. In this complex, H2A-H2B can be seen found between the importin and the Nap1 dimer. We also solved the structure of the nucleoform, the quaternary complex with RanGTP to 2.9 angstrom resolution. Here, Kap114, H2A-H2B, and Nap1 are arranged similarly as the cytosolic complex. In both structures, Nap1 and Kap114 together shield multiple nucleosomal DNA binding surfaces of H2A-H2B. Consistently, DNA competition assays show that Nap1 and Kap114 can effectively disaggregate H2A-H2B DNA complexes. We thus conclude that Nap1 and Kap114 co-chaperone H2A-H2B from aberrant interactions with DNA. However, we had previously shown that Ran binding to Kap114 H2A-H2B reoriented the histone, increasing its exposure to DNA and thus decreasing Kap114's chaperoning activity. We now show that the presence of Nap1 rescues the compromised chaperoning by shielding the DNA binding histone surface that was exposed by RanGTP. We also performed nucleosome assembly assays, which show that Nap1 histone Cap-Ran complex can effectively deposit its H2A-H2B onto tetrasomes. We further showed that a Kap114 mutant incapable of binding Nap1 released its associated histone in the presence of Ran and Nap1. These results suggest that within the quaternary complex, histone transfer likely occurs from Kap114 to Nap1 and then to the assembling nucleosome. To conclude, we found that Kap114 and Nap1 co-chaperone H2A-H2B while transporting it from the cytoplasm to the nucleus. Together, the importin and the histone chaperone provide a sheltered path to transfer H2A-H2B from Kap114 to Nap1, ultimately directing its specific deposition into assembling nucleosomes. We would like to thank the core facilities, collaborators, and funding agencies that enabled this study. Thank you.